All right, well, good afternoon, everyone, and thank you for joining us today for part 2 of our series on caring for cancer patients in the midst of the coronavirus pandemic. I'm Leah Streuber, and I'm here with our founder and chief medical officer, Dr. Ken Dixon, and we are so honored to have a very special guest with us today, Dr. Tony Letai. Dr. Letai, thank you. I'm glad you're here.
Good to see you, Tony.
So Dr. Letai is a medical oncologist and hematologist. He's affiliated with Dana-Farber Cancer Institute in Boston and is president of the Society for Functional Precision Medicine. And Dr. Letai, again, we appreciate your willingness to join the conversation today. And before I hand it over to Dr. Dixon, can you just tell us a little bit about yourself and your special interest in oncology?
Sure. I'm a faculty member at Harvard Medical School and Dana-Farber Cancer Institute. I have an MD, which means I have clinical medical training, as well as a PhD in molecular biology. And I've been working at Dana-Farber really my whole adult life. I did my training in internal medicine in Boston and oncology also in Boston at Dana-Farber Cancer Institute, and have stayed there as a faculty member. And what I realized is I became intrigued by the question of how do you know when a drug is going to work? And there was a particular drug I was interested in, venetoclax, and how are we going to figure out which patients it works in and which patients won't it work in? And it turned out the answers that popped up weren't genetics. It wasn't analyzing mutations. It was figuring out how the cancer cell worked and how the cancer cell worked differently from the normal cells. And that's what really put us on the track. And that's what made me think about trying to develop assays and methods that we could apply to clinical samples that really looked at the function of the cell and not just the genetics of the cell. And I also learned that we can apply these lessons, even though I started learning them with a drug called venetoclax, that they could be applied very broadly to many different kinds of drugs and many different kinds of cancer cells.
Tony, I'm going to shift a little bit of the direction. So in a minute, I'm going to ask you about how the COVID pandemic is affecting you guys in Boston. But would you talk for a minute about BH3 profiling and how you're looking at that as a way to determine when cancer cells are fixing to die after they've been treated with the drug?
Sure. So the first thing to explain is that it probably takes a lot of people by surprise that even though our body's made up of trillions of cells, every single one of those cells contains within it sort of a suicide program. It has within it, it's like instructions awaiting to be acted upon to commit suicide. Now, most of our cells don't act on those instructions. They sit around doing what they're supposed to do. But all our cells are made in such a way that if they go bad, if they do something just wrong enough, they activate that program and they kill themselves. And that cell is eliminated from the body for the sake of the entire body, for the sake of the healthy rest of your body. Those cells that are bad, maybe doing something they ought not to be doing, maybe growing where they shouldn't be growing, those cells are eliminated. Now that is regulated by this process called apoptosis. And this program called apoptosis is regulated by something that lives inside all our cells called mitochondria. You don't need to be a cell biologist, but mitochondria live inside cells, and if you've heard of them at all, you may have heard of them as producing energy. Our cells need energy to survive, and these are kind of like power plants for the cell. But they're also in some ways sacs of poison, so that if they are to burst, that's what triggers the cell to commit suicide. And what we did is we developed an assay to figure out how ready is that mitochondria and how ready is that to burst in a given cells. In some cells, it's really, really ready to burst. Those are the ones that we call like primed for death. They're really close to committing to death. And there's others where it's like not even close to bursting. And those are the ones that we consider really unprimed. What BH3 profiling measures is essentially how close a cell is to the edge of the cliff of committing to death, whether it's really close to the cliff or whether it's really far from the cliff. And it turns out by being able to make that measurement, we can make a lot of useful predictions about how to kill a cancer cell and what drugs will kill a cancer cell.
Thank you. We'll come back to that in a minute, but I wanted to ask you about this COVID pandemic and how it's affecting you, particularly in your practice, your lab, as well as the research in the clinical care community up in Boston.
So Dana-Farber, and although I spend most of my time doing laboratory work, I am also a clinician. And clinical work, I'm glad to say Dana-Farber and Brigham Women's Hospital has pretty much continued for the cancer patient unabated. So that any cancer patient who needs to be seen as an outpatient is being seen. Any patient who needs to be admitted to the hospital for emergent care of some kind, they get admitted and they're being taken care of. So the cancer patients haven't had a major interruption in their care. It is true, it's taught us, like many, to rethink how often a patient needs to come all the way into Boston into the clinic. And there has been an initiation of more telemedicine and via the phone as well as via contacts, just like we're having right here. So that I think that has changed the way some patients are seen. And it might be actually a more permanent change because there's definitely some advantages, especially when you're a referral center. Sometimes information can be exchanged that doesn't require an in-person visit. So we'll see how long that persists. So with regard to clinical care, I'd say it has not changed that much. There's been some influence on clinical trial initiation, but patients who are already on clinical trials have continued to get their therapy and so forth. With regard to research and laboratories, that has come largely to a complete halt. We were instructed to send all our laboratory personnel home so as to help out with stemming the infection. This came from Dana-Farber leadership, but ultimately city and state leadership also, of course, got involved, and people are not going to Dana-Farber to do laboratory work. To keep the laboratory engaged, we've been doing a lot of journal clubs, laboratory presentations, keeping ourselves intellectually engaged in the problem of cancer research, writing grants, and we've continued to write papers. We had, like many labs, we have data that is kind of backed up, and I've been able to personally use the time very productively, hammering away at my computer in this office that you see right here. The good news is I heard just today, just really within the hour before this started, is because not only nationwide, but also specifically in Boston and Massachusetts, we can now see a decrease starting, that we're on the downward slope, whether you're looking at COVID deaths or new infections or whatever measure you use, things are getting better. We were never overwhelmed like New York. It got pretty busy at one point, but never overwhelmed like New York. We're able to keep up with it. But now that we see the pace of infection coming down, things are going to be loosening up. So within the next week or so, the first kind of pioneers will be going back to the laboratory. And then after that, people will go back in shifts. It won't be the same as before. We're going to work it so that there will be fewer people in the laboratory at all times. People will be wearing masks, which they didn't do before. No doubt sort of hand washing and speaking and other sorts of contact we typically have will be changed for a long time. But we will go from having absolutely no lab work being done to, I'd say, probably something on the order of 50% productivity within the next few weeks.
What about you may not have this window into this world, but what about your surgical and radiation oncology colleagues there?
So I'm pretty sure RadOnc is doing what they need. Radiation oncologists are doing what they need to be done. Surgical oncologists, if like a resection is required, I think that's an example of the type of surgery that is still going on. And there have been emergent surgeries where surgeons have been extremely cooperative in the hospital. If someone had some sort of a bowel catastrophe while they're being treated with chemotherapy, they've been able to take care of those things and have been very helpful. However, surgical volume as a whole is down at least 90%. And so on the whole, the surgical colleagues and surgical patients have been very much set back by this whole COVID issue. Anything the least bit elective. This goes for the medical proceduralists too, like the endoscopists. All of those have been pushed into the future, and there's been a dramatic decline in the volume of any of these procedures, which has a significant effect on patient care as well as, of course, financial concerns for the hospital.
That meshes. Last week we had Clifton Hastings and Andrew Green, two of my local colleagues, and they were talking about everything's been shut down here, and we're probably opening up in the next few days to increasing surgical volumes. Let me shift over, Tony, and ask you about the inception of the Society for Functional Precision Medicine. You sort of alluded to it earlier, but this is congratulations on what I believe is a great innovation. But would you comment about that some?
Thanks. I mean, I guess it started, and I wished I could remember exactly who asked me this. Someone was asking what sort of research I did. I have this vague memory. It was maybe a family member, but it was a friend. It was someone who wasn't trained in medicine or science, saying, well, so we do cancer research. What do you do? Do you just like put drugs on cells and see if they work? And my response was something, very superficial. Oh, no, it's much more complicated like that, genes and molecules and proteins and all that business. But that stuck with me, and I wondered, I kept coming back to that in my mind. Well, why not? Why don't we do more of that? And I looked into it, and people had tried, and it hadn't worked very well. But really, that it was a process that made a ton of common sense, and we had much better tools than we did, say, in the late 90s, when it was largely sort of probably reached its apex, and then had declined since then. So I started to take it seriously. And then coincidentally, from some interest I had in trying to figure out how to use this drug venetoclax, the idea of just sort of putting the drug on cells, I did that. It worked great. And it was able to foster great predictions about where this would work in the clinic that all turned successful. And I said, okay, we need to seriously re-examine this issue of functional precision medicine, of putting the drugs we're interested in directly on the patient tumor cells. I mean, it makes so much sense. And then I started to discover that I wasn't the only one. I think it's a lot like you're I think in Georgia, so you may be familiar with The Walking Dead. It's sort of similar, like after the zombie apocalypse, there's these different groups that are still like around the world that are surviving. And I discovered that there were these different pods of scientists at different locations worldwide who had come to the same conclusion. But we were all operating very separately without talking to each other. And I thought that there were numerous reasons why it would benefit us to come together. So first, sponsored by the Forbeck Foundation, a very helpful foundation to me, they sponsored a meeting where a couple dozen people came. It was an international conference, and it was great. There was a lot of enthusiasm. It was wonderful to hear what everyone else was doing. And it was also wonderful to hear from a number of intelligent people that independently arrived to the conclusion that, yes, this is going to work, and this is something that's going to play a really important role in guiding patient care in the future. So after that, I decided, basically, well, one meeting is probably not going to be enough to move this forward, and what we really need is a society. So then I decided to found the Society for Functional Precision Medicine, and that was, I don't know, maybe 2 or 3 years ago, and now we have, I think, a pretty well-functioning society. We continue to grow, and I hope we continue to grow even after this. I'll point out to your audience, while we largely consist of scientists and doctors and people from academia and pharma, we're also very interested in patient advocacy and patient involvement. And if patients want to get involved with the SFPM, they can certainly go visit our website. You can contact us via the website, via the email there. We have a number of different portals on social media. We're very glad to hear from you. We'd love to hear patient involvement.
But yeah, Tony, as you know, our company SpeciCare is focused on collecting, saving, testing tissue of individual patients. It's an early movement, and you're spearheading that. We're trying to assist. Do you have any other comments about individual patients seeking out this new way of looking at things?
Yeah. So right now, the field is young. There are a couple commercial entities that are offering tests right now. I personally am not. We are not ready to do that. I would say, though, that your tissue is your own, and you get to decide what happens to it. And it is worth considering or discussing with your surgeon or oncologist. If you have a surgical procedure going on, to consider before you go in there, is there some of it you want to be tested in this way? And if there's a way to store it viably, that is, store it without killing the tissue. I think one of the things that we need to change very broadly is this idea that when you excise tissue from a cancer patient, really the first thing we do often is to kill all the tissue by putting it in a fixative like formalin or something like that. And all tissue biopsies start out live, viable tissue, and it's us who kill it. So we are actually inserting a step that interferes with the sort of functional testing that I think will eventually be very useful.
Very good. Well, I have a question for you, Dr. Letai. In several of our internal meetings and just in conversations, Dr. Dixon will occasionally mention something about poking a dog with a stick. And I actually understand that that's your story. So can you share it with us and maybe give us some points on what and on the points that you like to make from the story?
Sure. You can when you're trying to answer the question of if I put this drug on this cancer cell, what's going to happen? You can think of it as like the drug is sort of the thing giving a poke. And the cancer cell is a sort of complex biological system, just like a puppy dog is a complex biological system. So you can imagine if you're asking the question of what happens when you put a drug on this cancer cell, it's sort of like, well, it's how do we answer the question of like, what is that dog going to do if I poke it with this stick? Okay, how is that dog going to behave? And I like to contrast how a typical modern cancer biologist would answer that question with how a 6-year-old might answer that question. And I'll leave the audience to decide which one seems to make more sense to them. So the typical cancer biologist, if they saw a puppy dog sitting there and they wanted to know how it was going to respond if you poked it with a stick, the first thing they would do is they would kill the dog. Then they would take it and they would fractionate it into its DNA, its RNA, its proteins, its metabolites, and it would do some very complicated, very impressive modern procedures of measuring all these billions of molecules and coming up with long lists and arrays and Excel sheets and whatnot filled with information about all these molecules. And then this would all be fed into some sort of a supercomputer that would spit out information based on previous dogs that had been chewed up and analyzed in this way, how exactly it was going to respond if you poked it with a stick. So that's one approach.
Right.
That really dominates thinking about precision medicine and cancer nowadays. Those are the rules, okay? What this 6-year-old would do would really be considered cheating by a lot of modern cancer biologists. If the 6-year-old wanted to know what would happen to that dog if you poked it with a stick, what he would do is poke it with a stick. So that's what I think we should be doing we have the tools now. We didn't have them 30 years ago. We have the tools now to take patients' tissues, expose them to drugs, and get useful information. To poke the dog with the actual stick that you're doing it with. We don't have to go through all these shenanigans that sometimes are informative, but all too often, most of the time, are not informative in a useful way.
Very good story and practical application for sure.
Great. That's a great story. That's why you hear it about once a week, Leah.
There you go. That's good. Well, thank you for sharing it with us, Dr. Letai, and we do thank you so much for joining us today. That seemed to just fly by.
All right, thanks a lot. It's really been my pleasure.
Okay, so we actually, and for those of you that are watching, we do look forward to sharing more information about our company and our mission to offer patients customized cancer treatments on upcoming Facebook chats. We're going to continue to invite industry experts and offer relevant topics to keep you informed on industry trends. Be sure to check out our website at SpeciCare.com. Remember to follow us on Facebook, share us with your friends, and thank you again for joining this afternoon. Stay safe and have a nice day. Bye-bye.